So we are on chapter 3, biological macromolecules. The main student learning outcomes here is to understand the importance of macromolecules for living organisms. We will study how macromolecules form, either by a dehydration or condensation reaction or hydrolysis reaction. We will study carbohydrate structure and classification. And then we'll also study the basic carbohydrates like monosaccharides, disaccharides, and polysaccharides. We'll study the different lipids, its structure and function. Similarly, we'll study protein structure, function, and how proteins are organized, the different levels of organization of proteins. And we'll also study nucleic acids that's found in our body, deoxyribonucleic acid and ribonucleic acid, both its structure and function. An interesting way for us to understand the macromolecules is thinking about food. Everybody loves food. So you are what you eat. So whatever it is that you eat, our body will break it down. For example, the sugars, your bread, it will break down into glucose molecules to make energy. So you are, in other words, a turnover of what you eat. So you can think of the 4 macromolecules like how we approach food. So the 4, this is like the like a overview of what we'll be studying. There are 4 macromolecules: lipids, proteins, carbohydrates, and nucleic acids that make up all living things. For example, carbohydrate, the main function of carbohydrate is energy in the body. Yes, it's quick energy, especially glucose is quick energy. Yes. And in plants, like potato tubers, it will store it. So it also has a function of storage, and also it makes the cell walls in plants. So structure also. The unit that makes sugars, long sugars, would be called monosaccharide. So examples of monosaccharide is glucose and fructose. If it's 2 sugars coming together, it's called disaccharide. Thousands of sugars is called polysaccharide. So for the main macromolecule, here is the monomer or the units that make it up. For example, if you take lipids, it's not a monomer. There are 2 molecules that will come together to make the basic unit. Glycerol, the orange one is glycerol, and then there's a long chain of CH2s. So lipids are long-term energy storage molecule in your body. Like all the fat that we see in our body is for long-term storage. There's also a layer of fat under our skin, so it's a very good insulator, helping to maintain homeostasis in our body. The third group is proteins. Again, the function of proteins is just too big, diverse. The enzymes in our body are proteins. The antigen antibody or the immune system in your body is proteins. Muscle contraction, actin, myosin are proteins. Your hair is a structural protein, and so forth. The proteins are made up of monomers called amino acids. We'll talk about the structures soon. An example here is lactase, which is an enzyme. It will break down what? Lactose, and then into simpler units. Nucleic acids is the heredity molecule, the information storage molecule. The monomer is nucleotide. And then examples would be deoxyribonucleic acid and ribonucleic acid. So we'll move on to understanding how macromolecules are formed. So it's either through a dehydration reaction when they come together. So that's how molecules are formed. So for example, in the picture here, these are all monomers. Monomer means one molecule, one unit. So 2 molecules are coming together And with the loss of a hydrogen molecule, so in this animation it shows, a covalent will be formed. That bond is called, in sugars, it is called a glycosidic bond. So monomers will come together to form a long chain called polymer. Poly means more than one. And they come together to form covalent bonds. So in a dehydration or condensation reaction, so here in this example, two molecules are coming together to form, two molecules of glucose are coming together to form maltose with the loss of a water molecule. And the covalent bond that is formed is called glycosidic bond. The opposite happens in a hydrolysis reaction. So this is a long chain. It is a disaccharide. If you want to break this covalent bond here, the glycosidic bond, you have to add water to the system. So this is called hydrolysis. So hydrolysis reaction, here disaccharide maltose is broken down to form glucose monomers by the addition of water molecule. So they are kind of like the opposite. When you want to build something up, it is called condensation or dehydration reaction. When you're breaking down a large molecule, it is hydrolysis. So this is how all the macromolecules will form. Monomers will come together to form a large molecule by a dehydration reaction. So let's look at the first compound, which is carbohydrate. So every time we see carbohydrate, you write out the word. Carbo means what? Carbon. Hydrate means water. So it has 1 part carbon, 2 part hydrogen, and 1 part oxygen. So it's a 1-to-1 ratio. So the formula is CH2O, and then it can be several times, repeated several times. So the carbon of hydrogen, oxygen, and oxygen is a 1-to-1 ratio. There are 3 main examples that we'll be studying: monosaccharides, disaccharides, and polysaccharides. In your textbook, you would see a linear representation, structural formula, or like a cyclic representation. So you'll see both of them. If the carbonyl, see that group is terminal, it is called as a aldose. If it is found in the center of the molecule, it is called as a ketose. So the chain of carbon can either be 3. So then it is a triose. 5 pentose, hexose is a 6 carbon sugar. So again, the diversity that you see is immense. So monosaccharide is simple sugar, 1 sugar. So here's an example of glucose. See, another interesting thing is, see, glucose, galactose, fructose, they all isomers. They have the same structural formula, but the position of the different atoms are different. So they'll have different properties. They're structural isomers. Some good examples of monosaccharides would be glucose, fructose. They're all like sweet compounds. Fructose also because fruits are very rich in that sugar. That's why fructose. So the monosaccharide can either exist as a linear chain or a cyclic ring-shaped chain. Usually, the 5 or 6 carbon monosaccharide will exist in equilibrium between the linear and the ring forms. So, see, depending on the position of the hydroxyl group, see here in the carbon here, if the hydroxyl is down like this, it's called alpha position. Up, it is called beta position. So here's an example of a 5-carbon ribose sugar. Disaccharide is when two molecules come together, two monosaccharides come together by a dehydration reaction. So you can see glucose and fructose, they lose a water molecule, they form a glycosidic bond to form our table sugar. Again, very sweet. It's widely used in pastries and cooking. So di means two, saccharide means sugar. So there are two sugars. Another interesting thing you have to notice how the carbon is labeled. So you can see right next to the carbonyl group, you see carbon number 1. So it will be carbon number 1, 2, 3, 4, and so forth. So that's the naming of carbon. So again, the what is shown here is the same dehydration reaction with the loss of a water molecule. So the carbon atoms in the monosaccharide are numbered from the terminal carbon closest to the carbonyl group. So we have carbon number 1, 2, and so forth. So a few more examples of disaccharide. We already studied sucrose, which has one molecule of glucose and fructose. Maltose is glucose and glucose. Lactose is glucose and galactose. So they're all examples of disaccharide. Moving on to the large polymer, it is called polysaccharide. Polysaccharide. So that's maybe hundreds of molecules of glucose. So these are very high energy molecules. You can think of pasta that is or pizza, very, very high in carbohydrates. So if you take, say, starch, there are two main types of sugars, amylose and amylopectin. So polysaccharides are very long molecules, huge molecules that are linked together by glycosidic linkages. And some of the examples are starch, glycogen, cellulose, and chitin. If you take amylose, it's an unbranched chain. So the glucose molecules do not have side branches. They are unbranched chain and they are linked by alpha-1,4 glycosidic linkages. Whereas if you take amylopectin, that is shown in the bottom here, it is branched and it is connected by both alpha-1,4 and you can see alpha-1,6 glycosidic linkages also. So when you say 1,4, they're talking about carbon numbers, carbon 1 and carbon 4 of this next atom. Glycogen is the storage form of sugar in our body. So when there's excess glucose in our body will store it as a long chain carbohydrate, which is branched. So that is glycogen that you see in our muscles. So on a day when your body is low in energy, our body will take the glycogen reserves in order to make ATP. So in plants, plants do not have a skeletal system like us. And if you touch a plant, you know how stiff it is, right? Because of the cell wall. They have this special polysaccharide called cellulase. Cellulase. It is branched glucose polysaccharide. So the glucose monomers, what it will do is, it will form hydrogen bonds, forming these thick, very sturdy fibrils. So that is what we call as fiber. You eat salad, it's very good for our health, because it is full of fiber. But unfortunately, we do not have cellulase to break it down. But fiber is very important. It will keep your cholesterol under check. It is very important for bowel movements, right? To push the food down the intestinal tract. Because as fiber moves down the colon, it will tickle all the mucus producing cells. So that's why the food with peristalsis, which is the movement of food in your intestines, is very smooth. So fiber is very important in your diet. not only for digestion, but also to keep cholesterol in check and so forth. Lots of benefits for fiber. So that is the cellulose that is found in cell walls of plants. And this is because of hydrogen bonds between the chains, making these strong fibrils. Moving on to the next polymer, it is lipids. Think about the fat that's under your skin. It is for long-term energy storage. There's lots of energy in a molecule of fat. It is the insulator of our body. So it is a long chain of repeating CH2 units. And CH2, like long chains, are very, very hydrophobic because they are non-polar. They don't mix with water. And most of the common examples are fats, phospholipids, and steroids. So a lipid is not a polymer of monomer, but the monomer that makes a lipid is two molecules. That the top molecule is called glycerol molecule. And it can form covalent bonds, 3 covalent bonds with a long chain fatty acid. So that long chain fatty acid is that stretch of CH2s, full of energy. So a dehydration reaction will happen with the loss of 3 water molecules. These 3 fatty acids will bind with the glycerol molecule. So glycerol backbone. So here the covalent bond that forms here is called ester bond. And there's a very good example of this otter. You know, it has this hydrophobic skin that will repel water. That's why it has a good time in a water environment. The water is just going to fall off its back. The next thing for us to understand would be if a fat is saturated or unsaturated. When they say something is saturated, so like the molecule on top, they're saying it is saturated with hydrogen. So you can see there's only single bonds. It is full of hydrogen. If there is a lack of hydrogen, it will form a double bond. So unsaturated fats will have 1 or more double bonds. And it can either be in a cis configuration, see both the hydrogen are found on the same side, or in a trans configuration. But in the cis configuration, what happens is when there is a double bond, the shape of the molecule will change. So instead of the molecule being straight, it will form like a bend in it. There's a kink in it. And because of the kink, the molecules can't stack up on top of each other. So in a saturated molecule, if it is saturated fat, all the molecules will stack up on top of each other. So it is a solid in room temperature, like butter. Moderation is the key. Too much saturated fats can... Yeah, it stops your heart. So it can cause plaque in the arteries. It leads to cardiovascular disease. But if it's an unsaturated fat, right, it has a kink in it, the molecules can't stack up on top of each other. So it is a liquid at room temperature, like olive oil. That's why this is what's recommended. You know, all in right proportion though, because they are high calorie molecules. And of course, you know, in order to improve the shelf life, you can artificially add hydrogen atoms to something good like this and make it a trans fat. So again, that is also very bad for you, trans fats, like shortening. You know, they add it for cookies and cakes in order to keep it fresh for a long period of time. The next most important molecule that comes under lipids would be phospholipids. See, as the picture shows here, instead of one glycerol molecule having 3 fatty acid chains, and it's called triglyceride that we saw in our earlier slide, here, one fatty acid is replaced by a phosphate group. And because of that replacement, that molecule is very, very unique. It becomes an amphipathic molecule. So instead of being hydrophobic, you have the head of the molecule is water loving, hydrophilic, whereas the tails are hydrophobic. So the phospholipid molecule naturally forms lipid bilayers like this. So the heads are turned away facing the aqueous or water environment of the cell, and the tails, which are hydrophobic, are protected inside the bilayer. So the main concept that you have to understand is phospholipids makes all the cell membranes. It's an amphipathic molecule. Amphi means both. It has hydrophilic heads and hydrophobic tails. So it can form a lipid bilayer. The next group of fats and lipids is steroids. So it has a cyclic structure, as you can see here. So it's a 4-ring structure. Good examples would be cholesterol and cortisol. So they are very hydrophobic. Usually our liver, apart from the diet cholesterol, our liver can also make cholesterol. And it serves as a precursor molecule for important hormones like testosterone and estradiol. Also for making vitamin D and bile, that is important for emulsification of fats during digestion. Cholesterol is also a very important component of the cell wall. It will keep the cell wall very fluid. The next macromolecule is proteins. It is one of the most abundant molecules in our body. And it has like a series of functions. For example, all the enzymes in your body, here it mentions digestive enzymes like amylase that's going to break down starch, lipase to break down lipids, pepsin to break down proteins. So for all the digestive processes, these enzymes are important. Transport proteins in your body, hemoglobin that is found in your red blood cells. They will carry oxygen, carry substances in the blood and lymph throughout the body. They can be structural. For example, your hair is very rich, hair and nails are rich in keratin. Actin and tubulin form the cytoskeleton in a cell. The hormones in your body, like insulin that balances your blood sugar, is a protein. The immune system of your body, all your antibodies and immunoglobulins, they are all proteins. For movement, for contraction, actin and myosin are proteins. In eggs, the protein is stored as albumin. So this is like the short list of protein, the importance of protein in our diet. So what is the structure of proteins? The monomer that makes protein is called amino acid. So here is an amino acid. How many different types of amino acids are there? 20. You're right. And see, there is a central carbon. It is attached to an amino group. See that NH2 group, which is kind of basic in nature. And then it has a COOH group, which is acidic in nature. So it has an amino group, a carboxyl group, and it also has a hydrogen. So we have a central carbon. Remember, valency of carbon is 4. And then it can be attached to a R side chain. So that is the side group. So there are 20 different types of side groups. So that's why there are 20 different types of amino acid. And this will determine the property of the amino acid. For example, you can see glycine, alanine, valine, leucine, methionine, isoleucine, proline. They are all non-polar. And here's some examples of positively charged R groups like lysine, arginine, and histidine. Aspartame and glutamate are negatively charged. And then we have some non-polar R groups like phenylalanine, tyrosine, and tryptophan. Polar uncharged groups are like cysteine, serine, threonine, aspargine, and glutamine. So these are the 20 different amino acids, which are the monomers that will come together to form a long chain of protein called polypeptide. So again, they come together to form a covalent bond. And here the covalent bond is called as a peptide bond. And the long string of amino acid is called as a polypeptide. Protein structure is very, very complicated. So it has all these different layers of protein structure. We have primary structure, secondary, tertiary, and quaternary structure. And protein structure is very, very important for its function. So when they say primary structure, it refers to the amino acid sequence of the polypeptide chain. Secondary structure refers to hydrogen bonds that form between the carbonyl oxygen and the amino hydrogen in the peptide backbone. So because of this hydrogen bonding, the molecule can have a spiral shape. So it's called alpha helix, or it can take a sheet-like shape. So that is called beta pleated sheet. So different amino acids will have different tendencies to form these structures, either a spiral or a sheet. The next level of organization is the tertiary structure. So this is talking about hydrophobic interactions, ionic bonds, hydrogen bonds, or disulfide bridges that happen within the polypeptide chain. So the long polypeptide chain can kind of fold back on itself depending on all these interactions. And when we say quaternary structure, the fourth level of folding, we are talking about polypeptides that many polypeptides will come together to form one protein. For example, hemoglobin, 4 polypeptides have to come together to make the protein. So the interaction between these subunits is the quaternary structure. So for proteins, we have the primary structure, secondary structure, tertiary, and quaternary structure. And this is very, very important for its function. There is a phenomenon called protein denaturation. So this highly folded active protein, if it is subjected to like extreme temperatures or like chemicals of difference in pH, it can unravel like this. And that phenomena, when the protein loses its 3-dimensional structure and its function, it's called denaturation. Sometimes it's reversible, but many a time it's not reversible. It's like, cracking an egg on a hot skillet. The albumin protein in the egg is going to coagulate, right? So that is protein denaturation. Here's a very good example of why protein structure is very important. So this is sickle cell anemia. It is most common in certain races. So it's a point mutation in the DNA of hemoglobin. And because of that mutation, you can see in position number 6 in this picture, glutamate, instead of glutamate amino acid, it is replaced by valine. And because of that one change, right? Instead of being a nice round hemoglobin molecule, so that is the 3D structure of a hemoglobin molecule, the molecule will collapse on itself and become sickle shaped. And they are very sticky and they can form fibrils. So these sticky molecules will clog arteries and then it gives pain attacks. It's a very sad disease. So this picture is like showing you the importance of the structure of proteins. The last macromolecule is nucleic acids. When we say nucleic acids, it includes deoxyribonucleic acid and ribonucleic acid. The main concept here, why do they say it is the information molecule in your body? It is the inheritance or hereditary molecule in your body. Because the DNA that is protected inside the nucleus will make a molecule called RNA that will leave the nucleus. And then the RNA will act as a template for protein synthesis. This is called the central dogma of molecular biology. It's a powerful finding. Picture right there. So nucleic acids are macromolecules. The monomer that makes nucleic acid is called nucleotide. So a nucleotide has something called as a base, a sugar molecule, and a phosphate molecule. So that is the nucleotide. The bases can either be pyrimidines or it can be purines. Pyrimidines are single ring structures. Cytosine, thymine, uracil are pyrimidines. Adenine and guanine are purines. So if you take a nucleotide, it will either have a purine or pyrimidine. It will have a sugar, and then it will have a phosphate group. The pyrimidine uracil is found only in RNA, ribonucleic acid. It's not found in DNA. So these are the building blocks that make nucleic acids. So in RNA, it's a ribose sugar. In DNA, it is deoxyribose sugar. So you can see in position number 2, there is no oxygen. Position number 2, there is no oxygen in DNA. That is why it's called deoxyribose sugar, whereas it is found in RNA. So you have to know the basic difference between RNA and DNA. So this is the final slide. So here we have the beautiful double helix DNA structure, helical structure of DNA. So DNA is a long molecule. There are 2 strands. It looks like a twisted ladder. The sides of the ladder are the sugars and phosphates, bonded by covalent bonds, whereas the rungs of the ladder are the bases. And the bases will base pair in a very unique way. Adenine will always base pair with thymine, 2 hydrogen bonds, and guanine will base pair with cytosine. So that's what you're seeing there. So this is called DNA base pairing group. And then the 2 strands of DNA are not going in the same direction. They are going in the opposite direction. It is called anti-parallel. So for example, you can see in the diagram on the top, the free 5' end here, and here is the 3' end. It's called 5' and 3' end. So one side it's going from 5 to 3, the other side is going from 3 to 5. So it's the opposite. So it's called anti-parallel. And some of the main differences between DNA and RNA is, of course, the function. DNA is the master molecule. It is always protected in the nucleus of a eukaryotic cell like us. When it wants to express itself, it will make an RNA molecule, which is single-stranded. It will come out to the cytoplasm of the cell where it will act as a template to make proteins. We are going to study all this in the future lectures. Another difference, RNA has ribose sugar, DNA is deoxyribose sugar. So this is an overview of everything we'll be covering in the future chapters. And I hope that was helpful.